MTHFR reductase mutation means nothing relating to vaccines. So if you've been told that vaccines interact with the MTHFR enzyme, then that's a complete load of rot. If vaccines were filled with super high levels of homocysteine or folic acid or folate, sure, you should be concerned. But having one or two MTHFR gene mutations has absolutely no relationship with the vaccine schedule. None. If you've been advised by a witch doctor using the name of osteopath or chiropractor or medical journalist, or even maybe a nephrologist or someone who promotes chelation therapy for autism or some other woo practitioner, that there is some type of relationship between an MTHFR gene mutation and vaccines, then you've been lied to. MTHFR has absolutely nothing to do with vaccines at all. If you want more information, you can quickly go to omim.org, omim.org, and search up MTHFR. This is where you'll find not only the genetics of MTHFR, but every verified case control study confirming genotype, phenotype, alleles, loci, and related chromosomal abnormalities. Everything you want to know about MTHFR. So let's have no more talk about bogus MTHFR genes linked to vaccines. But for those who want the science info, I assume that you have at least a basic understanding of biology and more specifically genetics, and how haploid cells, one from your father and one from your mother, join together to become a diploid cell. Just taking you back, from these two haploid cells, the spermatocyte and the oocyte, you receive half your genes from your father and half your genes from your mother. At the point when the two haploid cells become a diploid cell and start the process of mitosis, these two lots of genes mold into one lot of genes, your genes, and every single cell in your body is produced from that point on, has those genes in it, all the good parts and all the bad parts that go into your genes from your parents at that point, at mitosis. Any mutations or changes in DNA also occur at that point when the two haploid cells become a diploid cell. So changes such as autism spectrum disorder and today's topic, MTHFR, they all happen at that point. So you may or may not have seen these words, but at a cursory glance, MTHFR may look like a cuss word, but it's not. It's simply a biochemist's lazy acronym for a gene that we all have. Everyone has this gene, and this gene encodes for an enzyme. So MTHFR stands for MethyTetraHydroFolate Reductase. There's so many bogus memes and bullcrap YouTube conspiracy theories amongst anti-vaxxers that certain health issues are associated with an MTHFR mutation and somehow it's linked to vaccines. The MethyTetraHydroFolate Reductase enzyme basically plays a role in amino acid synthesis. So if the MethyTetraHydroFolate Reductase enzyme is not functioning at full capacity, the patient may or may not have homocysteine blood level problems. MethyTetraHydroFolate Reductase takes a molecule that we call 5,10-methylene-tetrahydrofolate to a molecule called 5-methylene-tetrahydrofolate. This reaction is required for a multi-step process that converts the amino acid homocysteine to another amino acid called methionine. The body uses methionine to make proteins and other important compounds. Methionine is an amino acid. Amino acids are the building block that our body uses to make up proteins, such as methylene tetrahydrofolate. Methionine is found in meat, fish, dairy products, and it plays an important role in many cell functions. Methionine is also used as medicine to prevent liver damage in Tylenol poisoning. So non-functioning or low-functioning methylene tetrahydrofolate reductase can be monitored by blood tests. Blood tests can reveal hyper, homocysteine, high levels of homocysteine, or even low levels, hypohomocysteine. However, neither of these blood tests show the cause of the hyper or the hypohomocysteine, only the presence or concentration of high or low levels of homocysteine. Further genetic tests would show conclusively whether an MTHFR mutation is present or absent. I'll come back to the genetics. Let's continue on with hyper or hypohomocysteine symptoms. Doctors may order a homocysteine test if they suspect you have a vitamin deficiency and if you begin exhibiting symptoms of a vitamin deficiency. So whether you have the vitamin deficiency or you have the symptoms of the vitamin deficiency. So symptoms of a vitamin B12 or folate deficiency that thought to be MethyTetraHydroFolate Reductase diminished capacity include pale skin, weakness, fatigue, tingling sensations like pins and needles in your arms and hands, legs and feet, dizziness, and mouth sores. You also get mood changes, fatigue, and tongue swelling. There could be growth problems relating to vitamin deficiencies as well. So symptoms of vitamin deficiency anemia overlap with the B12 and folate deficiencies and also cause other symptoms and from anemia and that together include fatigue, muscle weakness, unsteady movements, pale and yellowish skin, personality changes, shortness of breath or dizziness, irregular heartbeat, numbness or tingling in the hands and feet, mental confusion or forgetfulness, weight loss, and so on. So there's many factors that contribute to high homocysteine levels. If you have a folate or vitamin B deficiency, you may develop hyperhomocysteine. This doesn't mean that you've got the mutation on the MTHFR gene. Other risk factors for hyperhomocysteine include low thyroid hormone levels, psoriasis, kidney disease, certain medications, and different genetics. So hyperhomocysteine itself usually does not cause any symptoms in adults, but it can in children. Symptoms can vary from person to person and be next to nothing or subtle. Depends on the person. A normal level of homocysteine in the blood is less than about 15 micromoles per litre, and up to severe levels goes over 100 micromoles per litre, but normal is about 15 micromoles per litre. Studies show that you can have either one or two or more standard mutations on the MethyTetraHydroFolate gene. Okay, so you can have none, 1, 2, 3, or more. These mutations are called variants, and a variant is just a part of the DNA of a gene that is commonly different or varies from person to person. So having one variant, it's called heterozygous, is very unlikely to contribute to any health issues, which means that you're heterozygous for the MTHFR gene mutation. It is believed that having two mutations in which you are homozygous for the MTHFR gene may lead to more serious problems. Two or more. So there are two main variants that if you Google it you'll find that occur on the MTHFR gene. And these two are usually listed in the documents. So the specific variants are C677T and A1298C, although there's a few more and I'll mention them briefly later. So 677T, approximately 30 to 40% of the population have this mutation at position 677. Approximately 25% of Hispanic people and 10 to 15% of Caucasian descent are homozygous, which means that they have two mutations for this variant. C677T is a base pair substitution. So from school biology or college, you may remember that cytosine binds with guanine, thymine binds with adenine. In the C677T mutation, CT, guanine has bound to thymine, hence the name CT. So This is a complete mismatch in code and can produce enzymatic functional defects. Can produce enzymatic functional defects. Did I say guanine binds to thymine? Cytosine binds to thymine at 0.677 on the methylene tetrahydrofolate reductase gene. The other common base pair substitution is A1298C. So in position 1298, there's an A to C bridge. So as I've earlier mentioned, cytosine binds with guanine and thymine binds with adenine. In this one, A1298C, adenine binds with cytosine, hence the name A to C. And there's another so this is another mismatch which can produce enzymatic functional defects. And notice again I use the word can, because there's not a does produce enzymatic functional effects by having this gene. So A1298C is an incorrect base pair substitution at position 1298 on the methylene tetrahydrofolate reductase gene. So around 20% of American population have this homozygous mutation at position A1298C. It's also possible to acquire both C677T and A1298C mutations and have one of each, okay, from your parents or from one or the other. Gene mutations are inherited, like I said, which means you acquire them from your parents at conception. You receive one copy of the MTHFR gene from your father and one from your mother. If both have mutations, your risk is higher of having a homozygous mutation. The chromosome cytogenetic location for the MTHFR gene is on chromosome 1 on the P arm and section or area 36.22. The methylene tetrahydrofolate reductase genes and phenotypes, they've been known to cause homocysteinuria due to MTHFR deficiency, and this is autosomal recessive. Susceptibility to neural tube defects, which is also autosomal recessive. Susceptibility to schizophrenia, which is autosomal dominant, and susceptibility to thromboembolism, which is also autosomal dominant, and you can be susceptible to vascular diseases with high levels of homocysteine. So there's some pretty big words in there, and if you don't understand metabolic pathways, let me break down what happens for you. You can also stop this at any time and Google. In your body, thiomethylfolate, which we call folate, from your diet or from the cellular folate pool stores in your body, by using serine to guanine in synthesis, converts thiomethylfolate to 5,10 methylene thiohydrofolate or folate. And then with the methylene tetrahydrofolate reductase enzyme, which is coded by your DNA from the methylene tetrahydrofolate reductase genetic region on chromosome 1, like I mentioned earlier, this methylene tetrahydrofolate is reduced, hence the word reductase, to 5-methylene tetrahydrofolate. So from 5,10 methyl tetrahydrofolate to 5-methyl tetrahydrofolate. And this is part of the synthesis of methionine to homocysteine. If there's a low level or no traceable level of 5-methyl tetrahydrofolate, then this severely restricts the synthesis of methionine to homocysteine. So what exactly is homocysteine? Do I need to go over it? Let's go over it quickly. Homocysteine. Homocysteine is a common amino acid found in your blood, and high or higher levels of homocysteine are a risk factor for heart disease. And with elevated levels of vitamin B6, B12 and folate, you can also have renal disease. Now for something really cool and handy and useful for life and knowledge. Have you ever heard of formaldehyde? Every anti-vaxxer's heard of formaldehyde. Well, this formaldehyde chemical, it's in the air. We breathe it in every day. We breathe in approximately 10 kilos of formaldehyde a year. So that's a lot of formaldehyde. It's also in the water we drink and in our food. So not to be outdone, in the air and the water and the food, vehicles without or with faulty catalytic converters also pump out formaldehyde. It is released from some brands of home insulation and from furniture and carpet foam cleaners. So this formaldehyde is everywhere. Trace amounts of formaldehyde are found in cigarette smoke, from forest fires, and from natural product decay. So our body also makes formaldehyde. 5,10-methylene-tetrahydrofolate is an intermediate in the detoxification of formaldehyde. This happens whether you have a homozygous or heterozygous MTHFR gene mutation. That happens anyway. So Goyette, in a study in 95, used reverse transcriptase PCR of RNA from MTHFR deficient patients, followed an analysis and identified 3 substitutions in the MTHFR gene, 2 missense mutations and 1 nonsense mutation. Why am I talking about missense and nonsense? The nonsense mutation and the 1 missense mutation were threonine to methionine. That pathway was identified in severe early onset patients. The second missense mutation, arginine to glycine, was identified in 2 patients with a thermolabile enzyme and late onset neurological disease. You can Google them if you want to know what thermolabile is. Frost in 95 identified a C to T substitution at nucleotide 677. I've already mentioned that. And that converts alanine to valine residue and is responsible for the synthesis of a thermolabile form of MTHFR. This is C677T, found by Frost in 95. Goyette, again in 96, reported an additional 5 mutations causing MTHFR deficiency. They also reported results of analysis of the enzyme thermolability in 22 patients with MTHFR deficiency. 6 of the 22 had 4 mutations in the MTHFR gene, 2 rare mutations causing severe deficiency, and 2 mutations for the common alanine to valine mutation associated with enzyme thermolability. Kutmans in 98 reported the molecular basis of severe MTHFR deficiency in 4 unrelated families of Greek and Turkish ancestry. By using reverse transcriptase PCR, they followed by direct sequencing and identified 4 novel homozygous mutations: 983A to G, 1027T to G, 1084C to T, and 1711C to T. 938A to G is an incorrect base pairing of adenine to guanine at position 938. 1027TG is an incorrect base pairing of thymine to guanine at position 1027. 1084CT is an incorrect base pairing of cytosine to thymine at position 1084, and 1711CT is an incorrect base pairing of cytosine to thymine at position 1711. They also found a splice variant containing a premature termination codon, probably as a secondary effect to 1027T to G missense mutation in 1 patient. Sibiani in 2000 stated that 18 rare mutations had been reported in patients with MTHFR deficiency. In addition, 2 well-known polymorphs have been found to cause mild enzyme deficiency. Mild. They reported 6 novel mutations, bringing the total to 24. They stated that each of the 22 patients were compound heterozygous for 2 separate mutations. So that's what they studied, people with 2 separate mutations. Sibiani in 2003 stated that 29 mutations in the MTHFR gene had been identified in homocysteine patients, as well as 2 common variants, alanine to valine and glutamine to alanine, in individuals with mild enzymatic deficiency. And here's the interesting part. Expression of 3 mutations showed a decreased enzyme activity between only 10 and 36% of control levels, with little or no affinity for 5-methylene tetrahydrofolate. Okay, low. Expression of the 3 mutations with the 677CT polymorphism also observed in the patients resulted in an additional 50% decrease in enzyme activity. So the more that's there, the more the enzyme activity will decrease. Tonetti et al. In 2001 reported a family in which 2 of their 4 children were affected by MTHFR deficiency. One of the affected children exhibited some clinical findings suggesting cystathionine beta synthase deficiency. MTHFR enzymatic activity was extremely reduced in this case. The mother was homozygous for the mutation on exon 9 and was clinically and bioclinically normal with normal folate status, although MTHFR activity was moderately decreased. She comes out as normal. The father was heterozygous for a transition arginine to stop codon and for the common polymorphism 677CT on exon 4. He exhibited major biochemical abnormalities, hypohomocysteine and low methionine levels in red blood cells, but he was clinically normal. The unaffected children had biochemical patterns close to that of their mother, and they were heterozygous for the mutation on exon 9, but also for the 2 common polymorphisms, 677CT and 1298 A to C. So in the affected children, some biochemical abnormalities, including folate status and especially methylfolate levels, were improved with treatment combining MethyTetraHydroFolate acid, hydroxycobalamin, pyroxidine, and betaine. However, homocysteine concentrations remained high, and methionine concentrations were lowered. In 11 families with the deficiency, Tonetti in 2003 identified 14 mutations, 10 of which were novel. They found no biochemical or clinical correlations with the mutations, none. Martin in 2006 undertook a systematic identification and investigation of common polymorphisms and haplotypes in the MTHFR gene, and using a genotype to phenotype strategy followed by functional genomic studies. Specifically, they resequenced exons and splice junctions and portions of the 5' flanking region of the human MTHFR gene using 240 DNA samples from 4 ethnic groups. A total of 65 polymorphisms were observed, 11 of which were non-synonymous coding, and we call that CSNPs or CSNPs. Let's quickly touch on population genetics so we know what's been studied. Wilkin in 2003 studied geographic and ethnic distribution of 677CT polymorphism in more than 7,000 newborns from 16 areas in Europe, Asia, the Americas, the Middle East, and Australia. The TT genotype was particularly common in northern China, 20%, southern Italy, 26%, and Mexico, 32%. There's also some evidence for geographic gradients in Europe, so north to south increased, and China, north to south decreased. Areas at the extremes of the frequency distribution showed deviations from Hardy-Weinberg expectations, namely Helsinki, southern Italy, and southern China. The findings suggested the existence of selective mitotic pressures leading to marked variation. Then Goyette in 1998 defined the structure of human and mouse genes. The human MTHFR gene is composed of 11 exons. The exon sizes, location of intronic boundaries and intron sizes are quite similar between the two species. They did mice and humans. Gorgon in 2000 analysed the promoter region of the MTHFR gene and found that it does not have a Tatar box but contains CpG islands, multiple potential SP1 binding sites, and binding sites for several other transcription factors. You can find all these things and much more if you go to omim.org, omim.org, and by searching MTHFR. Or you can type in the whole thing if you want. Research around MTHFR and its effects are still evolving. Evidence linking most of the health conditions that I've mentioned in anti-vax memes, or ones that are made up and listed in anti-vax memes and on YouTube videos to MTHFR, is either currently lacking or has been disproven time and time again, and should be taken with a grain of salt if you want to waste salt. More than likely, unless you have specifically diagnosed folate problems or have testing done, you'll never become aware of your MTHFR mutation status if you have one. Conditions that have been proposed to be associated with MTHFR by anti-vaxxers are cardiovascular and thromboembolic diseases such as blood clots, strokes, embolism, heart attacks, depression, anxiety, bipolar disorder, schizophrenia, colon cancer, acute leukemia, chronic pain and fatigue, nerve pain, migraines, recurrent miscarriages in women of childbearing age, pregnancies with neural tube defects like spina bifida and anencephaly. The anencephaly is the babies that have a decreased or missing cerebellum and top of their skull or head. The risk is possibly increased if a person has 2 or 3 or more gene variants for the MTHFR mutation. Having one MTHFR variant doesn't mean you need any medical treatment. It could just mean you need to take a supplemental vitamin B. It may not even mean that. It may not even be noticeable. Treatment may be indicated when a person has very high homocysteine levels, almost always above the level attributed to most MTHFR variants. Doctors should rule out other possible causes of increase in homocysteine, which can occur with or without MTHFR variants. So diet and so on, and lifestyle. Other causes of high homocysteine level can include hypothyroidism, conditions like diabetes, high cholesterol, high blood pressure, obesity and inactivity, certain medications, and such as fenofibrate and methotrexate and nicotinic acid, smoking. And from there, treatment will depend on the cause and doesn't necessarily take into account anything to do with MTHFR. The exception is when you have all of the following together. Tick the box. High homocysteine levels, a confirmed MTHFR mutation or 2 or 3 or 4, vitamin deficiencies in folate, choline, vitamin B12, B6 or riboflavin, complications in pregnancy, and recurrent miscarriages and neural tube defects. They're potentially associated with MTHFR. The Genetic and Rare Diseases Info Center says studies suggest that women who have 2 C677T variants are at an increased risk of having a child with a neural tube defect, but you need to have the 2 or more. A 2006 study looked at women with a history of recurrent miscarriages, and it found that 59% of them had multiple homozygous gene mutations, including MTHFR, associated with blood clotting, versus only 10% of women in the control category. You should, if you're pregnant, speak with your doctor about testing if you've experienced, or if you're not pregnant, if you're experiencing several unexplained miscarriages or have had a child with a neural tube defect, or if you know that you have an MTHFR mutation and become pregnant. You should talk to your doctor. Though there is little evidence to support it, some doctors suggest blood clotting medications, but there's nothing to support that. Extra folate supplementation may also be recommended, but may or may not help. Potential supplementation. The MTHFR gene mutation inhibits the way the body processes folic acid and other important B vitamins. I've told you, changing up your supplementation of this nutrient is a potential focus in countering its effects. Should talk to your doctor though. Folic acid is actually a man-made version of folate, a naturally occurring nutrient found in foods. Taking a bioavailable form of folate, so methylated folate, may help your body absorb it more readily. Most people are encouraged to take a multivitamin that contains at least 0.4 milligrams of folate or folic acid each day. Pregnant women are not encouraged. You're not encouraged to switch prenatal vitamins or care based on your MTHFR status alone, or your assumption that you have it. This means that taking a standard dose of 0.4 to 0.6 milligrams of folate daily should be satisfactory. Women with a history of neural tube defects should speak to their doctor for specific recommendations about nutrients. Neural tube defects can be caused by other factors other than MTHFR, so don't jump to that conclusion. Speak with your doctor before changing vitamins and supplements. Some may interfere with other medications or treatments that you're receiving, so don't just jump at extra supplements. Your doctor may also suggest prescription vitamins that contain folate versus folic acid. So in your diet, eating foods rich in folate may help you naturally support levels of this important vitamin. Supplementation may be necessary. Good food choices can include proteins like cooked beans, peas and lentils, veggies like spinach, asparagus, lettuce, beets, broccoli, corn, Brussels sprouts and bok choy, fruits like cantaloupe, honeydew, banana, raspberries, grapefruits and strawberries, juices like orange, canned pineapple, grapefruit, tomato or other vegetable juice, and peanut butter. Sunflower seeds are also high in this. So the takeaway from this, your MTHFR status may or may not impact your health. Genetic tests are the only tests that can confirm MTHFR status. If diagnosed with MTHFR variants, there are few possibilities and factors to consider. There's a few. MTHFR variants can be heterozygous or homozygous. MTHFR variants affect a few genes, mainly 677 and 1298 polymorphisms. Additional genes have been studied, but more testing is needed to know the effect that they may or may not have. And here's the kicker. If you have one gene mutation, either 677CT or A1298C, there's a very high chance that your MTHFR enzyme works okay and you show no signs or symptoms whatsoever. You have probably gone through life just fine without having high homocysteine levels or knowledge of it, a functioning folic acid pathway and functioning formaldehyde degradation pathway, and you're probably doing just fine and you haven't noticed. MTHFR gene mutations with 2 or more mutations inherited from your parents, C677 and A1298C, then yes, there may be folic acid or folate synthesis issues. If you're concerned because your homocysteine levels are high, talk to your doctor and have some tests done, some genetic tests. High homocysteine levels in themselves don't always guarantee a free pass to my child can't have vaccines because they have MTHFR mutation. That's a complete load of crap. If vaccines were filled with super high levels of homocysteine or folic acid or folate, sure, there's a reason to be concerned, but having 1 or 2 MTHFR gene mutations has absolutely no relationship with the vaccine schedule. Absolutely none. Having 7 MTHFR mutations has nothing to do with the vaccine schedule. If you've been advised by a witch doctor, an osteopath, a chiropractor, a woo practitioner like a lawyer or someone, that there is any relationship between the MTHFR gene mutation and vaccines, you've been lied to. MTHFR has absolutely nothing to do with vaccines. So if you'd like more information, you can find me on social media. Also talk to your doctor. Give them a heads up what you're coming to talk to them about so they can get some information for you. Keep in touch with medical professionals, people that deal with this every day. Okay, so let's have no more bogus talk about the BS MTHFR gene and vaccine link, because there isn't one. Bye. G'day, mate.